Friends, welcome back to my channel, and today in this video we are going to study about cytokine antagonists. Cytokine antagonists means these are molecules that reverses the effect of cytokines. First is interleukin 1 receptor antagonist. In short, we can write this as interleukin IL-1 receptor R antagonist. And from the name itself, we can understand that interleukin 1 receptor is antagonist to the cytokine interleukin 1. The cells that produces interleukin 1, the same cell also produces interleukin 1R. Right? Now, why this is needed? Interleukins, or in general the cytokines, these are the chemical messengers of our body. Now, because of these cytokines, different other cells of our body gets activated, and also it leads to inflammation. Now, when the amount of cytokines increases, then what is required for our body, it will create inflammation, more inflammation, and because of which it will start harming our own cells. So therefore, to stop the effect of excess of cytokine, we have the antagonist system in our body that helps to balance the level of cytokines. That means the amount of cytokines which is produced by our body should remain in a balanced form, just necessary to produce the signal or to kill any pathogens. And after that, after the work has been done, it should be stopped. And to stop that, or to stop the effect of excess of cytokines, our body also produced some antagonist. So one such antagonist is interleukin 1R, which is antagonist to interleukin 1. Okay. Now interleukin 1 helps to activate signaling pathway, whereas if it is interleukin 1R, it won't activate the signaling pathway, and that's how it stops or reverses the effect of cytokines. Now, under clinical conditions, we have one recombinant interleukin RA that uses for the treatment of rheumatoid arthritis. Rheumatoid arthritis is a type of autoimmune diseases due to which the person develops a lot of inflammations around joints. So that is rheumatoid arthritis. So excess of inflammation is because of excess of cytokines, and that excess of cytokine can be stopped by using interleukin RA antagonists. Now, there are some antagonists that are derived from the cleavage of cytokine receptor. That means the antagonists are a part of receptor itself. What do we mean by receptor? Cytokines are the molecules, and they activate the cell after binding with the receptors present in that cell. Such activation can be paracrine in nature. Paracrine means activating nearby cells, or it can be autocrine in nature, that means activating the self cell. So there are some antagonists that are actually the part of receptor itself. When they get cleaved and separated from the receptor, now this molecule, which was earlier the part of a receptor, will now function as an antagonist. Okay, one such example is of interleukin 2R, right, which is, as the name suggests, 2R. That means it is antagonist to the interleukin 2 cytokine. So interleukin 2R is a type of antagonist which is normally a part of the receptor to which interleukin 2 binds. When this part gets separated from the receptor, now this part will work as an antagonist for this interleukin. Okay, so this is one type where the antagonist is a part of a receptor itself. Now next is, apart from having all these protective system in our body, still we get attacked with the pathogens, still we get diseases. So there are viruses that exploit the cytokine activity to evade or to invade our body. Now what are the strategies that are used by various viruses? The first is generation of viral products that interfere with cytokine secretions. That means whenever we have a viral attack, our body will produce cytokines to call other cells of our body to that position so that the other cells can kill the virus. But some virus secretes some specific products, and the function of these products are they interfere or they stop the production of cytokine or they stop the secretion of cytokines. When cytokines is not secreted, that means the message is not available to other cells of the body that can come and inhibit the virus. So virus can easily invade our body. Now, second strategy is generation of cytokine homologs that compete with natural cytokines or inhibits antiviral response. Now, this is the cell which produces cytokines. Now, whenever any virus attack our body, they will produce similar molecules like cytokines. Now, say for example, T cell should come and kill the virus. Now, for that, the T cell has some receptors where this cytokine should bind. But because cytokine homologs are produced by the virus, this cytokine, which are actually the homologs, not the actual cytokine, will compete with the normal cytokine, and instead of normal cytokine, this viral cytokine will bind to this receptor. So once the viral cytokine is bound with the receptor, definitely gene expression won't take place and T cell won't proliferate, and that's how it cannot attack the virus. So virus is now safe and it can invade our body to cause disease. Next strategy is the production of soluble cytokine binding proteins. That means they produce some proteins that bind with the cytokines and thereby inhibit the binding of the cytokine with the receptor. Expression of homologs of cytokine receptors. Instead of producing the cytokines, there are viruses that produce the homologs of cytokine receptors. Next, the generation of viral products that interfere with intracellular signaling. The induction of cytokine inhibitors in the host cell. So, few examples are mentioned. For example, Epstein-Barr virus, which produces interleukin 10-like molecules. So, when it is interleukin 10-like molecules and not the exact interleukin 10. So, if interleukin 10-like molecules will bind with the receptor, what will happen? The cell won't get activated because it is not the actual cytokines. So, what is the function of interleukin 10? Th1 activation. So if it is bound with interleukin 10-like molecule, it won't activate Th cell. Okay. Similarly, pox virus is there, which encode soluble tumor necrosis factor binding protein and also interleukin 1 binding protein. So tumor necrosis factor binding protein. So these molecules will go and bind to the tumor necrosis factor. When they are bound with the tumor necrosis factor, it won't activate the cell that it normally does, right? So similarly with interleukin 1 binding protein. So interleukin 1 binding protein means now interleukin 1 is bound with some another protein. So when it is bound with another protein, it cannot bind with the receptor, and that's how it cannot activate the cell. Now this is a table which shows different viruses and what are the homologs or proteins they produces, and that's how they can invade our body. So that's all for this video. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for next video notification. Till then, stay safe, stay happy.